Hi everyone, my name is Joseph Maggiore and I'm really excited to talk to you today about the BioSystics Analytics Platform. So I'm an MD PhD graduate student in my fourth year at the University of Pittsburgh. I have no conflict of interest with the BioSystics team here. This is just kind of something that I've used in my own research and have worked really closely with the team in actually utilizing this. And as a future clinician scientist, really believe that this is kind of a necessary step, especially in the development of patient digital twin technology. So I wanted to start with just asking a question, and do you have a platform for a complete data pipeline from discovery to preclinical trials and patient digital twins? Because that's really what the BioSystics AP has as its mission, is creating kind of this one platform for that. And for those of you that are familiar, this is actually the commercialization effort that was formerly the MPS database system from the University of Pittsburgh, and this can now be accessed at the biosystics.com. So the BioSystics vision itself is creating this computational patient digital twin, and this really comes from kind of two data sources here. Patient clinical data, this can be EHR data, omics data, that's combined with patient-derived MPS data to inform this patient digital twin that is really the next generation of drug discovery modeling. So this is just a schematic of what the database actually looks like. It's an aggregation of preclinical data with clinical data and MPS data that can interact with public databases and proprietary databases that all generates this actionable knowledge in the drug discovery space. So when we're referring to MPS, we're really referring to these fit-for-purpose systems that can range, all the way from 2D static culture systems all the way to really complex integrated fluidic systems. And in order for this you-on-a-chip technology to really have full actionable potential, it really relies on patient-derived stem cells in that it allows for the creating of individualized organ systems that can then inform precision medicine and a precision drug therapy application. So the BioSystics AP itself, it's available online, and it's also available for an installation behind a firewall. So there's a nonprofit version that is for academic and other nonprofit institutions, and there's also a commercial version for companies that are developing or using MPS experimental models. And I just want to kind of emphasize that the data that goes into the BioSystics AP, the data providers themselves actually have complete control over how their data is actually shared. So it can be, protected completely behind a firewall, but can also be shared, with collaborators or even to the greater research community itself. So this has been an effort for the past 10 years or so, and it's amassed 5,000 users that have contributed, almost, over 500,000 data points, which is a really massive amount of data already. And it's a number that has been, that grows week by week. So what I wanted to do is actually show what the BioSystics AP actually looks like. This is the actual graphical user interface. And you can see that there's, this area for actual study design where you're actually creating individual studies. But then those same studies can actually be implemented and then analyzed all in this same analytics platform. So this is what the workflow actually looks like, going from study design and implementation to actually bringing the data into the analytics platform. For modules like statistical analysis and computational modeling, all that can then be shared within your control. So what I wanted to do now was kind of walk through a couple of the different features and how, some actual case examples of what it actually looks like to use some of these features. So what you're seeing here is actually a comparison between a 3D MPS liver model and a 2D MPS liver model, and looking at albumin expression. So what's interesting about this is every line that you're seeing is actually a totally, novel liver model that is being studied and is being compared on the same graph. So here you can actually be modulating, specific parameters or specific outcomes to see how a given finding changes over time and across multiple studies. So this can be actually integrated to understand from a reproducibility angle how reproducible a given finding is. So you can kind of get this green flag, yellow flag, red flag metric. That's not an end-all be-all, but is kind of a next step to understand which findings are most reproducible in which specific conditions. There's also statistical modules for actual power analyses that can be used before an actual study is executed or even after to understand the statistical relevance of a given finding. Like I mentioned, the AP can interface with public databases and also proprietary databases, and what I'm showing here is a comparison of FDA adverse event reports normalized to CDC usage for a couple of different drugs that can really be well used for almost a positive or negative control for toxicology studies. So here you can see tolcapone has, an order of magnitude more adverse events than, a negative control like acetaminophen. The AP is also great for analyzing omics data sets, especially in a setting where some of these analyses require a computational background. Here they can kind of be uploaded and easily interpretable, but also be used to actually generate a mechanistic insight. So I'll show an actual case use of this, but here you can easily click and understand for a given cluster of RNA-seq data what are the actual upregulated genes and understanding what kind of pathways are upregulated for that gene ontology set. The AP is also really great for not only just image storage and video storage here, but also for quantitative analysis. So what you're seeing here are control and then troglitazone treated and understanding transporter efflux inhibition. And this is quantification that you can actually do within the BioSystics AP. The last I'll mention is there's a variety of computational modules that are already available, like this one here, which is the pharmacokinetic parameter prediction module, that actually can be used on experimental data that you may already have, or even experimental data, or historical data rather, that can be really helpful for then interfacing with other computational models or future modules that the BioSystics AP is going to integrate. So like I mentioned, I'm a graduate student at the University of Pittsburgh. I'm in my 4th year, and my main work is in developing human kidney organoids. And what I wanted to do was kind of first talk about some of the case usages of how I use human kidney organoids, and also how I've used the BioSystics AP to really advance some of our work. And then also some future applications in the digital twin sphere that are related to the human kidney organoids, but also in the greater MPS sphere as well. So this is what our group, Dr. Neil Huckread's group, does. We work with iPSC-derived human kidney organoids, and we have a couple of different mechanisms for injuring these kidney organoids to understand acute kidney injury and chronic kidney disease and the progression between these two. And we use them for 2 different focuses, being drug discovery focus and also trying to gain mechanistic understanding of the disease progressions. So what I personally see is, even in this group that I work in, even in an academic setting, is there's a massive lab-wide need for the management of this data. I'm generating a lot of data, and there's also years of historical data of people who have done experiments in our group. So not only is it important to just have some space to actually store all this data and to be able to share it within the lab and within lab collaborators, but also to be able to actually aggregate the data to compare, okay, I'm doing one study right now. Did someone 5 years ago also do this study, and are we getting a reproducible finding from 5 years ago? And lastly, to be able to not only just compare these studies, even a statistical way, but also to understand a mechanistic insight around this, to generate graphs that actually can help create a hypothesis around a question that we can then study in a future experiment. So I wanted to show some of the actual results and what it looks like in the AP itself from our human kidney organoid work. So this is modeling injury, and in the red you see the injured group, and in the blue is control. And here's a whole slew of qPCR markers from just one kidney organoid assay, and I can easily view this and see, and actually view it even with my PI, which is a great resource that we can sit together with actual graphs and not just a PowerPoint slide, and also have it be directly linked to the raw data itself. And we can see specific findings, like here HEMOX1, this is a really important kidney injury marker, and we see that after just 1 day of injury, there's a high upregulation of this injury marker. We can also look at a couple different injury markers at a time, and more importantly, look at multiple kidney organoid assays to assess, is this even a reproducible finding over time? So this is 3 to 4 to going up to 10 kidney organoid assays, plotting them all on the same graph to understand how reproducible something is. And it's not even, we've actually used this mostly in the setting of understanding how reproducible a finding is even between cell lines. So you can see here, when we look at some of these injury markers with one cell line, we get this, quote, acceptable reproducibility output. But using a completely different cell line, we see that there's actually a poor reproducibility. And not that this is, oh, we're never going to use this cell line again, but actually we can expand this out to understand, okay, maybe it's actually one assay that's really throwing off the reproducibility, and when we look at the specific protocol from that one assay, maybe we made one specific adjustment, and all of that is logged already within this kind of the standardized input for the analytics platform. So as I've mentioned, we also explore RNA-seq data within the BioSystics AP in a really easy to use. And what we're looking at is the human kidney organoids that have been injured over time. And this is great just to be able to plot this, which would be a normally computationally intensive process. But we also use it to actually generate hypotheses. So like I mentioned, if we were really interested in the progression of AKI to CKD, and we have hypothesized always that there's this epithelial mesenchymal transition that occurs in this progression. And when we looked at just one of these clusters in the RNA-seq dataset in the BioSystics Analytics Platform, we were able to quickly see what the upregulated genes actually were, and they were able to be exported into Enricher. And we saw that one of the top upregulated gene ontology pathways was the epithelial mesenchymal transition. So this was a really great way and kind of surprising finding that corroborated one of the hypotheses that we had from data that we already had in a way that we could kind of as a group sit together and look at raw data but get an actionable answer around. So what I want to just do in the latter half of this really is talk about some of the evolving applications in using patient digital twins. So this is kind of specific to kidney organoids, but really generalizable to any kind of MPS. And talking about how in any of these three applications, it really requires an analytics platform that can kind of amalgamate all of this because it's such a massive amount of data, whether it's MPS development or precision medicine or even drug discovery. So I'll kind of walk through each of these, starting with MPS development. So even in our own group, as we are generating, using more and more different cell lines for high throughput MPS generation, it's kind of, we're pitting this against what we feel is almost this, discordant data set of, at University of Pittsburgh, we have so much EHR data and patient data, and it's so important as we're developing these MPS to understand that this is actually a clinically relevant model in these, what seems to be disparate data sets. So it seems pretty necessary to have a kind of analytics platform that from the beginning of gathering this data can really start to synergize this and draw correlations between the MPS that's being generated and the clinical outcome. There's also great relevance in the precision medicine space, so generating patient-derived iPSC lines and generating multiple different MPS models, and not only being able to use some kind of analytics platform to compare the different MPS models themselves, but to use each of those in a high-throughput drug screening context to understand, okay, maybe this specific MPS works much better for this specific disease application and this specific set of compounds, in conjunction with some of the PK modeling that exists in the BioSystics Analytics Platform to say, okay, for this patient, this is actually the best drug that can be used for this specific clinical setting. And that clinical setting can be extrapolated even to some more of the drug discovery applications, specifically in understanding some patient heterogeneity. So, we're all well aware of how much variety can actually exist in a global patient setting. And as MPS are actually generated with all of this heterogeneity encompassed, with an omics approach, there's really three critical analyses that we think that are really possible with this patient digital twin application. In understanding actual target pathways, so with all of this data combined, what are the key pathways that are critical to these specific diseases? Not only that, from an actual clinical setting, maybe there is a small subset of this massive patient data cohort that actually is specifically best responsive to a specific drug and maybe based on a specific clinical phenotype. And finally, with biomarker identification, in summating all of this data into one actionable platform, how can we actually identify, are there maybe specific predictive biomarkers that inform a patient disease progression. So, from my perspective as a future clinician scientist and an MPS developer, it really seems like for any of these analytics platforms, this is kind of, for any of these applications rather, it kind of requires an analytics platform almost like yesterday. The data is constantly being generated for this, but it needs to be going in a format that can be constantly creating actionable knowledge as the data is actually accruing and synergizing it with actual clinical data for a patient digital twin application. So just a couple other points. Again, I use this analytics platform right now. It's available to log in. Feel free to provide any feedback as you're actually putting data in and trying it out. The team actually also wanted to acknowledge that, as you may know, starting in 2023, there will be a requirement for data sharing policy from NIH-funded projects, and the team is happy to talk with you about kind of creating a plan for that, since if the data is going to be shared publicly, it may as well have actionable answers that come along with that. There are webinar series that are readily available through the help menu on the analytics platform that I've used in the past. And I think most importantly, I've scheduled these interactive sessions with the team before to actually help inputting the data so that I can really get quickly to the actionable answers that we're trying to get. And the team I'll be at the booth as well. It's booth 123, and the team is there to schedule these kind of interactive online sessions. So with that, I just want to acknowledge the team that, over the past 10 years has really put in a massive amount of work to bring this analytics platform to thousands of users currently and hopefully more in the future. Thank you.